we're the newest group in TSL. My group just moved in in October, this month, in October 2020, but we're all very excited to start doing something new in collaboration with other colleagues. One is the effector evolution. We identified these tandem repeats, and each unit we think is a structural and functional module of effectors in Phytophthora pathogens, and this is probably the most devastating plant pathogens that impacting many crops and forest trees too. And so we found several hundreds of these effectors that carry these modules, but they are in slightly different sequences, and the number of repeats are also different. And we see a lot of mosaic effectors that with these different sequences shuffle. So we're really eager to test whether the shuffling of these units confer different functions to these effectors, and how fast do they evolve. So we're sequencing and analyzing many Phytophthora genomes, looking for these tandem repeat containing effectors, and see their evolutionary dynamics, and also we characterize their functions during infection. So this is one focus. Another focus, the other focus is the small RNA in plant immunity. So we wanted to look for this specific pool of small RNAs and their evolution dynamics. Because if we consider small RNAs as a defense mechanism or antimicrobial agents, they're in constant battle with the pathogens, and the pathogen, the target sequence of the pathogen could change to escape that small RNA regulation, and on the contrary, the small RNA sequences should also change to counter that counter defense. So we do observe this very fast diversification of the small RNA producing sequences in Arabidopsis thaliana, which is the model plant species, and we wanted to expand that concept to other plants. And the third, but not least, project, we have been working on citrus Huanglongbing or citrus greening disease, and this is really the most devastating disease for citrus. And if we don't have a solution, we're at the edge of not having any juice for your breakfast. So this is a very severe disease, and it's a bacterial disease that colonizing the phloem tissue of the citrus trees, and they're transmitted by insect vectors. So we are interested in understanding how this three-way interaction happen, the insect, the plant, and the bacteria, and what's special about the phloem tissue to allow the bacterial colonization, and what happens in the bacteria, what are the virulence factors in the bacterial pathogen that can colonize the phloem and cause the disease. So we're really looking into the molecular basis of this this complicated pathosystem, but also very important disease, and we're hoping to contribute to the management of this disease in the near future. So we work on quite basic science, and if when we talk about our projects, and many of them, we keep saying molecular mechanisms. So what's the immediate impact, or how do I want to impact our society by doing our research? I think for sustainable agriculture, if we're talking about sustainable agriculture, we have to go down to mechanisms. We cannot just simply doing something quick and fast and with the hope that this would have a long-term solution. And I think we have learned that for a long time that sustainability requires a thorough understanding of this pathosystem, and we need to pool multiple defense mechanisms together to get to that disease management. So I think the research that we do in the lab will get that knowledge, will get that mechanistic insight that will definitely give us guidance to develop the sustainable disease resistance strategies, approaches. So for example, for citrus HLB or Huanglongbing research, we have identified this family of proteases that are in the phloem and targeted by pathogen effectors, and has been shown in other plants confer resistance to other pathogens. So I think this is a very clear target that if we can reinforce this defense, we may be able to better defend, protect citrus trees from the bacterial infection. So I think this is just one example of how our science could help the society. And I think the other thing that we shouldn't underestimate is the education part. I think, and I'm very happy with one of the missions of TSL is to not only translate the knowledge to the real practice, but also educate, mentor the next generation scientists. I think that's, and I'm very keen to also be involved in that. And I think the science, this young generation is our future. And if they can take good training from all different aspects, from ethics, from critical thinking, from knowledge, technology, different aspects of doing science and knowing that, and then they can go out and do science-related careers. And from their contribution to our society, I think we also fulfill our contribution to move the society forward.